Hi there, I'm Brianna Zarati, and today I will be presenting on my manuscript about predicting platinum resistance in ovarian cancer using baseline tumor RNA sequencing and machine learning. For this presentation, we're going to go over a few of our main sections. We're going to begin with our background, move on to questions of interest, then methodology, findings, and finally our conclusion. So we will begin with our background. So a little bit about ovarian cancer as a whole. Ovarian cancer is the 8th most common cancer and a leading cause of cancer-related mortality among women worldwide. In the United States alone, it was estimated that around 21,000 American women would be diagnosed and about 12,700 would die from ovarian cancer. And this is based off of statistics from the National Cancer Institute. One of the major challenges in ovarian cancer management is that early symptoms are typically nonspecific. So many patients do not get diagnosed until they're at advanced stages where those tumors have spread beyond the ovaries. As a result, treatment outcomes remain poor for many patients despite there being large advances in surgical and chemotherapeutic strategies. One of the treatment options for patients who have ovarian cancer is platinum-based chemotherapy. This includes drugs such as cisplatin and carboplatin. These remain standard first-line treatments for ovarian cancer following surgical tumor removal. These drugs exert cytotoxic effects by forming intrastrand and interstrand DNA crosslinks that interfere with DNA replication and transcription, ultimately triggering apoptosis in cancer cells. Despite initial responsiveness for many patients, many ovarian tumors eventually develop resistance to this platinum-based chemotherapy. This leads to disease recurrence and decreased survival. Clinically, treatment response is commonly assessed using the platinum-free interval, or PFI, defined as the time between completion of platinum therapy and disease reoccurrence. Tumors that relapse within 6 months are generally classified as platinum resistant, whereas tumors that recur after 6 months are considered platinum sensitive. And then any patients that have a shorter PFI typically experience poorer clinical outcomes and fewer effective treatment options down the road. You can see there on that left-hand side graph just the survivability. In blue we have our platinum sensitive folks, and in green we have the platinum resistant tumors, and you can see there's a vast difference in survivability between those two groups. The biological mechanisms underlying platinum resistance are really complex and involve multiple cellular processes. Proposed mechanisms include altered drug uptake and efflux, increased DNA repair capacity, dysregulation of apoptosis pathways, and interactions with the tumor microenvironment. Because these processes involve coordinated activity across numerous genes and pathways, identifying a single or even many biomarkers capable of predicting treatment response has proven to be very challenging. And with that, let's talk about our questions of interest. So the objective of my project here, and what I'm really trying to ask, is whether baseline tumor RNA sequence profiles that contain transcriptomic patterns associated with platinum response status and platinum-free interval in ovarian cancer can become a predictive model for other baseline tumors before standard treatment even begins. So by performing exploratory analysis of publicly available gene expression data, I really hope to identify global transcriptional patterns that may reflect biological mechanisms underlying platinum resistance and inform predictive modeling strategies. So let's move on to the methodology used to achieve this. So gene expression data and clinical metadata were obtained from the Gene Expression Omnibus dataset GSE214302. This dataset contains baseline tumor RNA sequencing profiles from ovarian cancer patients that have been treated with platinum-based chemotherapy. Gene expression values are reported as transcripts per million, or TPM, along with clinical variables describing patient age, tumor stage, platinum response status, and platinum-free interval. The RNA sequence TPM matrix initially contained expression measurements for about 59,609 genes across 18 tumor samples. To reduce noise and improve interpretability, genes with low expression levels were filtered prior to analysis. So genes with TPM values greater than 1 were in at least 20% of samples were retained, resulting in a filtered data set of 30,222 genes. Expression values were then logged to transformed to stabilize variance and reduce any skewness. Gene expression values were then centered and scaled to ensure comparability across samples prior to any type of dimensionality reduction. And then here is a look at that clinical metadata that I had previously spoken about. Here you can see that the median age was 50, and then the median PFI was around 15 to 16 months. With this, there were 10 platinum-sensitive and 8 platinum-resistant tumors that were retained for the analysis. So to understand the data set a little bit better, I decided to do a principal component analysis of the baseline tumor gene expression, as well as replicating the same thing but with the platinum-free interval for my exploratory data analysis. So the first principal component, PC1, explained 25.6% of the total variance, while the second principal component, PC2, explained 11.4%. Together, these two components captured a substantial portion of the transcriptomic variability that's present within the data. So figure 1 on the left-hand side shows the PCA plot colored by platinum response status. Partial separation between platinum-sensitive and platinum-resistant tumors is observed along PC1, with resistant tumors tending to cluster toward one side of the axis and sensitive tumors toward the other. However, no notable overlap remains between the two groups. Instead, resistance appears to arise from different complex and heterogeneous gene expression patterns across tumors. So to further assess the relationship between transcriptomic variation and clinical outcome, PSA results were visualized using a continuous color scale representing platinum-free interval right there on the right-hand figure. So figure 2 reveals a gradual gradient along with PC1, where tumors with shorter PFI values tend to cluster toward one end of the axis, while tumors with long PFI values appear toward the opposite end of the axis. And then to better understand which genes contributed most strongly to the observed transcriptive variation with the principal component analysis, we examined PC1 more closely. So the top genes contributing to PC1 exhibited similar loading magnitudes, indicating that no single gene dominated the primary axis of variation. This pattern suggests that global transcriptional programs, rather than individual genes, drive the primary axis of variation across tumors. So this figure here displays the top genes ranked by their contribution to PC1. The relatively uniform loadings across genes suggest that PC1 reflects coordinated expression changes across many transcripts rather than any isolated gene effects. And then finally, for our methodology for the predictive model that was built, so firstly I used lasso logic regression to evaluate the potential of individual genes as predictors of platinum resistance. So a binomial generalized linear model with L1 regularization was fit using gene expression values as predictors. The optimal regularization parameter was selected using a 4-fold stratified cross-validation. So to capture coordinated gene expression patterns, correlation-based hierarchical clustering was performed. Pairwise gene-gene correlations were computed, and a distance matrix was derived from these correlations and then used for clustering. The beginning resulting dendrogram was partitioned into 5 clusters, grouping genes with similar expression profiles across samples. For each sample, cluster-level expression scores were calculated as the mean expression of all genes within each cluster. This yielded a reduced feature set representing aggregated transcriptional activity. Cluster-level expression scores were used as predictors in logistic regression models to classify platinum resistance status. So models were fit using a binomial generalized linear model framework. Finally, model performance was evaluated using leave one out cross-validation, and classification accuracy was quantified using error rates. The discriminability of the models was assessed using receiver operating characteristic or ROC analysis, and the area under the curve was calculated to summarize the performance across classification thresholds. And with that, let's move on to our findings. So here are our results for our lasso cross-validation curve. So at this level of penalization, all gene coefficients were shrunk to 0, and only the model intercept remained non-zero. This resulted in no genes being selected by the lasso model for inclusion as predictive features. And this makes sense if we go back to our principal component analysis. We were showing that there were no huge standing out biomarkers or genes or anything like that. It was assumed that there would be more of a correlated effort between different transcriptional factors that would have an effect. And so this graph right here also is showing kind of a similar thing. So the feature selection failed to identify any individual gene as a predictive feature. This result reinforces the idea that no single gene strongly distinguishes platinum response in this data set. However, when we look at our correlation clustering, we see something a little bit different. So in contrast to the lasso model, clustering-based feature modeling enabled the aggregation of gene expression into biologically meaningful groups. The improved performance of the cluster 3 model suggests that these aggregated features better capture any underlying transcriptional programs associated with treatment response. So notably, cluster 3 contained a large set of genes, 272, and these genes contributed collectively to the predictive signal, including genes such as STAR D3NL, SF3A2, and others. These included both non-coding and pseudogene transcripts. So these genes span many different functional categories, including transcriptional regulation and RNA processing, and this suggests that both coding and non-coding regulatory processes may contribute to the observed differences in platinum response. So the results demonstrate that while individual gene predictors were not identified through lasso, coordinated multi-gene expression patterns captured through clustering approaches were associated with treatment response, with the cluster 3 model achieving a strong discriminative performance of around 0.888 for the ROC curve, as you can see there on the right-hand side. And these results make sense. These results align with prior literature demonstrating that platinum resistance in ovarian cancer is governed by complex, multifactorial biological processes, including alterations in DNA repair, pathways, apoptosis, and other things. Additionally, previous genomic studies have similarly reported that resistance phenotypes are driven by coordinated pathway level dysregulation rather than single gene biomarkers. And now, despite the strong performance observed in the ROC analysis for the cluster model here, these results should not be overstated. So in the beginning, we talked about how there was 18 samples, which is not a lot by any means. So that definitely limits the stability and the generalizability of the findings. With such a limited number of samples, model performance estimates, particularly those derived from cross-validation, may be optimistic and sensitive to minor variations in the data. So as a result, this should be viewed as a preliminary estimate rather than a definitive evidence of predictive accuracy. So finally, let's talk about our conclusion. So overall, this study demonstrates that baseline tumor RNA sequence profiles contain meaningful but distributed signals associated with platinum response in ovarian cancer. Exploratory analyses revealed global transcriptomic patterns linked to treatment response and platinum-free interval, while predictive modeling showed that cluster-derived multi-gene features outperform individual gene predictors. This was once again found through lasso, reinforcing the importance of coordinated transcriptional programs in driving resistance. Although cluster 3 model achieved a strong performance, these findings should still be interpreted cautiously, once again due to that small sample size and lack of external validation. Overall, I really think this work highlights the value of systems-level approaches for modeling complex biological phenotypes and provides a foundation for future studies aimed at improving prediction of chemotherapy response. And I hope in the future I can take the same methodology and expand it out to a much larger data set.